What do you think of when you hear the word cancer? Do you think about a diagnosis that destroys lives, a disease of the old, a swollen organ, or something that results in losing hair? Astonishingly, 40% of the population living in the United States will develop cancer during their lifetime. My name is Eric Stricker, and I am a PhD candidate at Baylor College of Medicine. And my goal today is for you to comprehend what cancer is, the factors that cause cancer, so that you can understand the different tests that can be performed to diagnose cancer. So let's have a look at the definition of cancer. Cancer is a genetic disease in which some of the body's own cells grow uncontrollably and potentially spread to other parts of the body. It usually starts with a single cell that is genetically altered, that divides more rapidly than normal. The cell will very quickly become a lump that becomes very solid, and then eventually turn into cells that dissociate from this different cell dense mask, leading to a malignant tumor, so spreading into other parts of the body, also called metastasis. The very cell dense body itself can be often seen in different scans, such as a mammogram for breast cancer. Here's a graph showing all cancers combined between 2013 and 2017 in their incidence rates, meaning how many people per 100,000 persons will be diagnosed with cancer. You very quickly see that over 50% of all cancers are diagnosed in people over the age of 70. But the question remains, what causes cancer? Is it artificial sweeteners, or is it weed killers, as we heard in the news? Is it UV radiation, or even smoking that causes cancer? Let's have a look at the observations from Dr. Alfred G. Knudsen in 1971. He observed that children with a family history of retinoblastoma often had both eyes affected. In contrast, children without family history often developed retinoblastoma only in one eye. So his hypothesis was that 2 mutations are required for retinoblastoma. I've drawn out here the different cells that we could find in the eyeballs, and assuming that these mutations are spontaneously occurring, we will find some mutations in different cells, as marked here. Now, the chance of 2 mutations that he postulated to be in the same cell is fairly low. For this reason, we will find cancer cells only in one eye, leading to retinoblastoma and sporadic retinoblastoma. In the case of familial retinoblastoma, we have one of the mutations inherited to every single cell of the body, including all the cells in the eyeball. It becomes very quickly obvious that when we have every single cell affected by the first mutation, the chance of a second mutation to occur in different cells is much higher. Therefore, we will find cancer cells more likely in more than one eye, leading to retinoblastoma in both eyes. The multiple hit hypothesis, on the other side, or also called multi-step model, is an extension of the 2-hit hypothesis. Mutations in several genes are usually required to produce cancer, which gave rise to the multiple hit hypothesis. These can be genes that affect cell growth, supply of blood to the cells, ability to spread, like in metastasis, and many more. Now that we know about the multiple hit hypothesis, we can start to distinguish sporadic, or also called spontaneous cancers, from cancer predispositions. Sporadic cancers develop without any family history, meaning there is no mutations present in the cells that make it more likely to develop cancer. That leads mostly that the cancer arises in later ages. If a cancer mutation is inherited, fewer somatic mutations are necessary for cancer to develop, and a person may have a predisposition to cancer. Very prominent example for that are leukemias and lymphomas, which are the most common cancers in children, as well as retinoblastoma that we learned about. So let's have a look at what kind of mutations lead to cancer. There are two major genes affected by mutations in cancer cells. On one hand, we will have the tumor suppressors. They are active at checkpoints and working like a brake. They inhibit in generally proliferation if something goes wrong in the cell, such as a DNA break or cells being infected by viruses. And they will inhibit for a certain time the cell growth till the repair is complete. And if the repair cannot be completed, will lead to the cell death. On the other hand, mutations affect proto-oncogenes. Proto-oncogenes are like the gas pedal in a car. They accelerate the normal cellular functions and control it, as well as lead to cellular growth, also called proliferation. They drive this in order for normal healthy cells, especially stem cells, to divide and supply the amount of cells we require on a normal basis for our body. Proto-oncogenes are very important, for example, for the renewal of our skin cells or liver cells that continually have to be renewed. Now, when we think about the mutations, there's a major difference between the two. And I want to just draw this out for you to understand. So imagine we do have a brake pedal. And we do have a gas pedal on the other side. Imagine, and I know this is not very realistic, we do have both feet on the brakes. Now, in order for the brake to be released, we would not only have to take one foot off the brake, but both feet. This is the reason why when we look at tumor suppressors, very often it requires mutations in both chromosome sets or in both chromosomes for a tumor suppressor to lead to a cancer, meaning that these tumor suppressors are usually recessive in their inheritance pattern. Now, having a look at the proto-oncogene, imagine we do have 2 feet on the gas pedal. Generally, when you drive, you don't press down the gas pedal completely. So in the same way, our 2 feet will have a well-regulated depth of gas pedal press here. Now, a mutation very often causes for the foot to press very deeply on the gas pedal, meaning that proto-oncogenes turn into oncogenes that have an active form that is not inhibited at all. Now it becomes obvious that only 1 foot, or let's say a foot of somebody else, was required to press on the gas pedal to accelerate us into a wall or into a way that is not in a good position. Accordingly, proto-oncogenes, when mutated into oncogenes, are inherited in a dominant pattern, meaning that only one part of the chromosome has to be affected. Usually the oncogenes will have a gain-of-function mutation that makes them continuously or constitutively active. When we look at the tumor suppressors, on the other hand, as I mentioned, we have a recessive inheritance pattern that usually leads to a loss of the function. Individuals that are heterozygous for the tumor suppressor genes are often predisposed to cancers. And there is this specific situation when a loss of heterozygosity, so if we lose one of the chromosomes in a single cell, it is more likely, or it increases the chances that there is a development of cancer. In order to better understand the diagnostic tools used to identify cancers, I wanted to look at the variety of different strategies that ensure the survival and growth. These are called hallmarks of cancer. These strategies can be divided into 6 general hallmarks, 2 emerging hallmarks, and 2 enabling characteristics. Diagnostic tests make use of these hallmarks to detect cancer cells. So let's have a look at the different diagnostic tests. So the first thing that a doctor suspecting cancer will ask you is about your environmental risk factors. They will ask questions like, do you smoke? Do you drink alcohol? Do you live in a very old house? And all these environmental risk factors increase the chances of developing cancer by causing continuous inflammation, or also called chronic inflammation. A chronic inflammation can lead to the inactivation of tumor suppressors through DNA methylations, for example. And DNA methylations are changes that block the activation of a gene and are also called epigenetic changes. Environmental risk factors for cancers are, like I mentioned, smoking, alcohol use, asbestos, but even high sugar diets can lead to increased risk. So looking at the hallmarks, we see that these risk factors can lead to a tumor-promoting inflammation. Usually followed by the environmental risk factor questions are questions about the family history. Often inherited cancer risk factors include the defects in DNA repair machinery. With an impaired DNA repair machinery, mutations accumulate quicker in all cells, increasing the chances of cancer mutations. So let's have a look at an example here. The BRCA1 and BRCA2 genes are important tumor suppressors responsible for the repair of DNA breaks. Specific variants of the BRCA1 and BRCA2 are known to increase the risk of breast cancer and are evaluated in genetic tests offered by companies like 23andMe. BRCA1 and BRCA2 are inherited in an autosomal dominant fashion despite being tumor suppressors, and are also found very often mutated in breast cancer, where they get their name from. So when the BRCA1 or BRCA2 genes are not working correctly, the DNA repair cannot work correctly, and it is more likely for an individual to actually develop breast cancer. So when we have a look back at the hallmarks of cancer, we see that mutations in DNA repair genes, for example BRCA1 and BRCA2, will lead to increased mutations as well as genome instability. Let's have a look at what genome instability looks. So when we compare a normal cell's genes and chromosomes to a tumor cell, we see that there's many different changes in the chromosome number and structure that are associated often with cancer. While a normal cell has each chromosome twice, with the exception of the X and Y chromosome, we see different changes, and I want you to take a second and maybe look at what different changes you can find here. So when we have a look at chromosome 3, we see that it's present 3 times, which is called a trisomy. While when we have a look at chromosome 5 or even 8, we see that we have only 1 copy of these chromosomes, which is called a monosomy. Marked here in the bottom are chromosomes that are altered. We see that the chromosome 2 has an extra region or also a duplication, while we see here in the 5th column a translocation. A translocation means that different chromosomes combined with each other to form a merged or chimeric chromosome. This can lead at the overlap to really problematic situations that we will have a look in a second at. And when we have a look at the chromosomes 9, 11, and 12 here at the end, we see that some of these chromosomes are actually lacking a majority of their genetic information, making them much shorter, which is caused by a deletion. So let's have a look at a specific example of a cancer caused by a chromosome translocation, which is chronic myeloid leukemia, also called CML. Chronic myeloid leukemia is caused by a gene BCR on chromosome 22 when it combines with the gene ABL on chromosome 9, resulting in a BCR-ABL fusion gene. A resulting new chromosome is called actually the Philadelphia chromosome, and is the main reason for CML to arise. The BCR-ABL fusion protein is a constitutively, which means permanently, active tyrosine kinase, which sends sustained proliferation signals, meaning the cells to divide and grow, and is immune to cell death signals, which in other words means that it evades apoptosis. Very interesting about this is that blood tests as well as bone marrow biopsies can especially useful to diagnose leukemias and lymphomas through tumor markers such as BCR-ABL fusion gene. So generally, a doctor would order a complete blood count, and when that appears to be normal, there is different tests that can be performed on the blood sample, such as an immunoglobulin test, a blood cancer antigen test, and circulating or even a circulating tumor test. For leukemias and lymphomas, we actually see the cancer right in the bloodstream as the affected cells are blood cells, and therefore we can test the tumor cells directly by looking at their genome and identify this BCR-ABL fusion gene, which is the cause. So when we have a look at the hallmarks, we see that BCR-ABL fusion proteins are an example for sustained proliferative signaling as well as resistance to cell death. In addition, many cancers are caused by defects in the cell cycle's regulatory machinery. The cell cycle itself just describes the different phases of cell growth and DNA synthesis, which are important for cells to divide and proliferate. The cell cycle itself is controlled at different checkpoints, which are only passed through activation of the correct cyclins and cyclin-dependent kinases, called CDKs. These are specific proteins that alter in their expression during these different phases and are essential for a control of the growth of the cells. Mutated genes in cancers often disable the cell cycle checkpoints. So when we look at the most frequently mutated gene, which is p53, which is also called sometimes TP53, for example, it is 75% of the time mutated in colon cancer. In normal cells, p53 is upregulated upon DNA damage, leading to the induction of the cyclin inhibitor P21. P21 then inhibits cyclin E and CDK2, arresting the cell and preventing it from growing till the DNA can be repaired. When DNA repair is not successful, the cell eventually will induce apoptosis, which basically means that it will lead to cell death. Loss of the tumor suppressor of p53 leads to an evasion of this growth suppression and resistance to cell death. While many tests can suggest cancer, a biopsy is the only way doctors can make a diagnosis. The uncontrolled growth of cancer cells will give them the distinct appearance. So continuous DNA replication will lead to large, darker, and variably shaped nuclei. When we have no cell cycle inhibition, like we just saw for p53 mutations, we will see many dividing cells with variable sizes. When we have a loss of DNA repair, we see abnormal number of chromosomes, like we saw for the BRCA1 and BRCA2 genes. And when we look at the activation of invasion and metastasis, which we'll cover in a second, we see clusters of cells which will have no boundaries. So going back at these different hallmarks, we see that a loss of the cell cycle control, for example by p53, can lead to the evasion of growth suppression and resistance to cell death. Now I want to talk about two aspects that are made use of for the diagnostic method of CT scans. Oxygen and nutrients are essential for the rapidly dividing cancer cells and are provided by new blood vessels. The creation of new blood vessels are also called angiogenesis. Genes that induce angiogenesis are often found overexpressed, while inhibitors of vascularization are mutated in cancers. The second aspect is that in the progression of tumor cells, cancer cells will lose their attachment to surrounding cells, which will then enter the bloodstream and spread to distant sites. The ability to penetrate to tissues is called invasion, and the formation of these secondary tumors are called metastases. CT scans use sequential X-ray images combined with a tracer that visualizes area of high blood flow. The tracer in the bloodstream and intestines blocks the X-rays, resulting in white areas. Now, when we have a look at this example, we see that the primary and metastatic tumor will appear white due to high vascularization, which is used very neatly by the CT scan. So having a look at the hallmarks of cancer, we can see that a CT scan can visualize induced angiogenesis as well as the activated invasion and metastasis. CT scans are often combined with PET scans, which stands for positron emission tomography scans. PET scans make use of the fact that cancer cells in particular depend on high amounts of blood sugar for energy, which is also called glucose. In the 1920s, Otto Warburg showed that cultured tumor tissues have high rates of glucose uptake and lactate secretion, even in the presence of oxygen, meaning that they perform anaerobic glycolysis even in an aerobic environment. This effect was called the Warburg effect. PET scans make use of the Warburg effect in cancer cells by injecting a traceable glucose, which is marked by positron emission isotopes, into the bloodstream and then measure the areas of increased signal. Cancers will light up very bright, giving high signal and showing a large glucose uptake. This is also a hallmark of cancers, as cancer cells are usually deregulating the cancer energetics to maximize their growth and proliferation. Genetic information is generally stored in form of double-stranded DNA in human cells. Double-stranded DNA itself is made out of two single strands combined in two opposite directions. While one of the strands can be replicated upon cell division in a continuous level, resulting into the leading strand, the opposite strand has to be replicated in fragments, called the lagging strand. The starting point of each replication is called the RNA primer, which later during replication will be replaced by DNA material. This is generally not a problem except for the end of one of the strands, which results into a gap when the RNA primer cannot be replaced by DNA. This generally results into a DNA strand that gets shorter with each application cycle. This serves as a timekeeper for cells, which means that when the ends get too short, the cell dies. In stem cells, which replicate much more often, for example in our skin, our intestine, or other organs, there is a specific enzyme called telomerase. The telomerase has an associated RNA that complements the 3' overhang at the end of the chromosome. The RNA template is used to synthesize the complementary strand, meaning the opposite strand that was parental. And telomerase shifts and the process is repeated, leading to these single strand gaps to get increasingly longer. Another enzyme called primase and DNA polymerase have the RNA primer that I mentioned earlier bind again, synthesizing the lagging strand, but this time in a much longer way, resulting in ends that don't get shortened. Telomerase is normally not expressed in somatic cells, meaning in normal cells, giving them a finite number of DNA replications. In cancer cells, often though, the telomerase is mutated so that it then is expressed, enabling replicative immortality. Now, looking at the hallmark, we see exactly that activating mutations in the telomerase gene can lead to the enabling of replicating immortality. As a last point, I wanted to mention a Nobel Prize that actually was given out to one of Houston's own researchers. So to avoid an immune response, tumor cells often upregulate immune checkpoint genes, which are the brakes of the immune system. Immune checkpoints such as the PD-L1, PD-1 pathway inactivate killer cells. Immune checkpoint inhibitors that were recently discovered are anti-PD-1 antibodies, for example, and can release the brakes from the immune system, enabling T cells to eliminate cancer cells. And in 2018, James P. Allison from Houston and Tasuku Honryo were awarded a Nobel Prize for their discovery of immune checkpoint inhibitors. Now, this completes the cycle of our cancer hallmarks with immune checkpoint molecules, which lead to the avoidance of immune destruction. I hope you enjoyed this small video about cancer genetics and learned what cancer is, what can cause cancer, and what diagnostic methods are used to identify cancer. If you have any questions, don't hesitate to reach out to me. With that, goodbye and see you next time.